I am Vaibhav Srivastav from Vaibhav E-Learning Academy, and today, after a so long time, we are going to discuss about an interesting topic in biology, and that is about blood groupings. So, we know that there are different types of blood groups, and we have usually heard about the different kinds of blood groups like A positive or B positive. But if we take the blood of individual people and if we examine it superficially, the blood looks the same. Then how are we able to determine which type of blood is positive blood group or which blood is an A positive or B positive? So for that, we are going to understand the whole detail about how can you classify a blood on the basis of what categories are we, or the basis of what criteria are we able to differentiate the blood between different types of groupings. And we will also understand what are the positive and negative meaning in the blood groups. Like we have heard about A positive and A negative. What is positive and what is negative? So in this video, we will try to understand all of that, and a part of this will be in the next video, that is about erythroblastosis fetalis, that is a medical condition, and we will be discussing that in the next video or a continuation of this video. So let us start. In today's video, we will be learning about the basics of blood, and then we will be learning about the blood grouping. So before moving on to the actual concept of blood grouping, we have to understand a few terms which are related to the blood. So we will be first knowing some basics about the blood. So let us start with the basics of blood first. So in our body, the most abundant fluid which is used for transportation of substances or gases is the blood. Now, this blood contains or consists of two important parts. One is called the plasma, and the other part is known as the formed elements. Now, in this video, every concept will be unwinding slowly. So, if there are some concepts which you do not understand in the starting, wait for some time because the concepts will be repeated in this video multiple times. Now, formed elements and plasma. Now, many people do not understand what are formed elements, but many of us have heard the term plasma. So, our blood contains 55% of plasma and the remaining 45% of formed elements. Now, these formed elements comprise of the most heard cells, that are the RBCs, the WBC, and the platelets. The RBCs are the red blood cells. The other name of the red blood cells is erythrocytes. Erythrocytes is the other name of the RBC, that is the red blood cell. WBC is the white blood cell. And the other name of WBC is leukocyte. The term leuco means colorless. So as there is no color of WBC, we attribute it to usually white. And then platelets, that are known as thrombocytes. These are the scientific terms of these cells. Now the function of RBC is to transport the oxygens from one place to other place, that is usually from the lungs to the tissues or back to the lungs. This RBC is red in color due to the presence of a very popular and very known protein or color pigment that is known as hemoglobin. Now if you have saw the video about biomolecules, then I have said that hemoglobin is made up of 4 quaternary structures, which is 2 alpha and 2 beta. Now white blood cells, WBC. So the function of RBC, as said, is the transportation, and RBC is the most abundant, that is 5 to 5.5 million per millimeter cube. So if you take 1 millimeter cube of blood, in that you can find 5 to 5.5 million or 50 lakh to 55 lakh million, 50 to 55 lakh RBC per millimeter cube of blood. WBC's main function is protection, or we can say response to any pathogen or response to any foreign microorganism. White blood cell has the majority function of white blood cell is protection or immunity. Now we can study detail about WBC in any further future videos, but for now we have to stick to the basics. WBCs are not as much abundant as the RBCs, but they are found 6,000 to 8,000 per millimeter cube of blood. So their main function is immunity. And then we have platelets which help in blood clot. And these are 1.5 to 3. 1.5 to 3 lakh per millimeter cube of blood. This was the basics of blood. Now, to understand the blood grouping, we need to mainly focus on plasma and the RBCs, because they will have some substances which help in identification of the blood group, and or which helps in the transfusion, that prevents the transfusion of the blood. So we have learned about RBC, and this is the thing about RBC. Now coming to plasma. Plasma is nothing but 90 to 95% water. And this 90 to 95% of plasma is water, and the remaining 2 to 8%, like it is usually 90 to 92%, and almost 8% is protein. If you want, you can get this as 92 as well. So we have almost 8% of plasma as protein. It also has some electrolytes, it means it also has some minerals, it also has some ions, and it also has some gases. So plasma is nothing but, from the whole blood, if you remove all these cells, the remaining liquid part of the blood is known as plasma. And the blood is liquid, right? So that liquid is because of this plasma. And this plasma is colorless, or you can say it is pale yellow color. There is a well-known term that is known as serum. Now what is a serum? To understand serum, we need to understand what are the proteins present in plasma. In plasma, there are 3 main types of proteins. I'll explain that orally. One is called as albumin, one is called globulin, and one is called as fibrinogen. Albumin protein helps in the maintenance of osmotic balance between the capillaries and the tissues. We will understand everything in detail in the upcoming videos. But the next protein over here is fibrinogen. Fibrinogen protein helps in the blood clotting. Fibrinogen protein along with the platelets perform a series of reactions which help in the blood clotting. And then we have globulin proteins. Globulin proteins are the proteins which help in immunity. They also help, they act with the WBC and help in the immunity functions of the body. Now there are these 3 major proteins present inside the plasma. Now what is serum? If from this plasma, if we remove the clotting factors, then it is called as serum. So if we remove the clotting factor inside the plasma is nothing but the fibrinogen. If we remove fibrinogen protein from the plasma, then the remaining part is known as serum. And usually whenever there is a need of blood to an individual who has amount of the cells are available, but if you need to transfuse some amount of liquid to a person, then usually serum is transfused, because we have to remove the clotting factors. If you remove the clotting factor, then there will not be any coagulation between the liquid of the individual who is donating and the individual who is receiving. This was the main base which we created about the blood. Now to understand the blood grouping, we have to understand plasma and RBCs. So now let us discuss about the blood groupings. So now let us understand about what is the main difference between an antigen and an antibody. Now these are the 2 terms which are very important to understand the blood grouping. So actually this video is about blood grouping, but we haven't even spoken about blood grouping till now. The reason is we are setting up the base to understand the blood grouping efficiently. Otherwise I will have to tell you the same things while explaining the blood grouping. So it's better to create a base first and then we'll understand the blood grouping. Now we have to understand what is the difference between an antigen and an antibody. If you do not understand this right now, it's not an issue, because while we are understanding the ABO blood grouping, or when we actually get into the concept of blood grouping, we will again try to relate the concept. So first let us create a small base about what is the difference between antigen and antibody. Now if you talk about antigen, if you break up this word, anti means antibody and gen means generator. So antigen is nothing but a substance that can generate antibody. Not a substance but or a site that is present on some cells which can generate antibody. And then the second one is antibody. This antibody searches for the antigen to destroy it. Now, to understand the concept of antigen and antibody, let me give you a small resemblance or a small analogy with the concept of vaccine. Now, how does the vaccine actually work? Now, in the process of vaccination, we inject a very weak or a dead pathogen inside a person's blood. Now, that pathogen will actually have its antigen. This antigen is a substance or a site which can define its identity, that this pathogen has this kind of antigen. Now, our body, that when we have spoken about the WBC side, our body produces antibodies in response to this antigen. So as we said, antibody is something that will destroy the antigen. So our body produces antibodies. These antibodies will destroy the pathogen's antigen. Now, once these antibodies have destroyed the pathogen's antigen, this antibodies get stored as memory. Since these antibodies have destroyed the antigen, and how were they able to destroy this antigen? Because this antigen was already either dead or it was very weakened. So it was easy to destroy it. This was like a drill for the body, that how can we prepare if any pathogen which has this antigen affects us. So these antibodies which are prepared by the body as an immunity response to this pathogen's antigen, this is stored as memory in our body, so that in future whenever any actual pathogen which is a strong pathogen which has this antigen enters the body, our body will easily be able to fight that pathogen's antigen, and kill it without much problem. So this is the concept of vaccination. Otherwise, if you inject a pathogen which is strong inside a body, then the body might show some reactions and it might take a long time to develop the antibodies and fight that pathogen. And by that time, the pathogen will have shown its effect. So this is the concept of vaccine. From here, we get an idea about what antigen and what antibodies are. Antigen is something that identifies, that is like an identity to a cell, that this cell has this kind of antigen. And antibody is the substance which can destroy the antigens. Now, usually our body does not produce the antibodies to the antigens which our body cells have. If our body cells are having, suppose, an antigen X, then our antibody will not produce anti-X, because this X is useful to us and it is in majority to us. So our body won't produce any antibody that is against this antigen, because if it produces, then our body might get in trouble. So this was a base which has been created. Now there are many points which I've said in this topic that many people do not understand, and that is why they do not understand the gist of the chapter. So what they have to do right now is, you have to just wait patiently, so that when I explain you the blood grouping, you will be able to easily understand what I mean by antigen and antibody. Now let us start the actual concept of blood grouping in the human. So now let us enter the actual concept of the blood grouping. Now, if you have noticed the blood grouping of some individuals, the blood grouping comes out to be as A positive or B positive, A negative, AB positive, O positive, O negative. These are the major blood groups which we have. There is also B negative. So, the blood group of an individual comprises of two terms. One is the alphabet, that is A or AB or O, and one is the positive or negative sign. So, from this we can conclude that in majority of the humans, blood grouping can be done in two ways. Or blood group is written by taking two grouping methods. One is called as ABO grouping, and one is known as RH group. Now, this ABO grouping will define the letters or the alphabets which are used in the blood group, and the RH grouping will describe or will define how and when to use the plus sign and how and when to use the minus sign in any blood group. So, let us start with the first one and the most easier one, that is the ABO grouping. Now, to understand this ABO grouping, we need to draw a table. So, let us start with the drawing of table. So now let us enter the main concept of blood grouping about the first one, that is ABO grouping. Now I have a table over here which will be filling in this video. So here in the table we have these four columns. One is the group, one is the antigen, one is antibody, and one is donor. So I have already given you an open idea about what an antigen and what an antibody is. So this antigen is found on the RBC, on RBC's surface. Suppose this is the RBC. If this is the RBC, then there is an antigen present on the surface of the RBC, which can tell which group the blood belongs to. Then there is an antibody. This antibody is present in the plasma. This is present in the plasma of the blood. So that is why I have given you the main basics about what is blood. This RBC is in a capillary, or it's in a blood vessel, and this surrounding liquid in the blood vessel is plasma. So antibody is a protein, or it is usually a protein which floats inside the plasma and it keeps on searching about the antigen. It searches the antigen which it has to destroy. So this is the main overall view about what the antigen and antibodies are. Then we have this donor. This donor is nothing but the people or the group which can donate blood. So we'll be even understanding who can donate blood to whom in this table. So there are 4 major groups: A, B, AB and O. So if you see the blood group of any individual, there are these 4 blood groups usually. There are also different types of groupings such as Bombay grouping as well, but that are exceptional cases and there are very rare cases. We'll be discussing about the major and the important one. So there are 4 groups: Group A, Group B, Group AB and Group O. Now Group A has the antigen of A. Group B has antigen of B. Group AB has antigen A and antigen B. And Group O has no antigen. None. So now we can understand how are we naming the blood groups. We are naming the blood groups on the basis of their antigen. The blood group A has antigen A, the blood group B has antigen B, blood group AB has both antigens A and B, and blood group O has no antigen. So suppose if I draw RBC over here and that RBC has an antigen which I name it A, then this blood group of this, the blood group of this individual will be A. If I have a RBC over here which has both A and B antigen, then this individual's blood group is AB. So this is the main concept of how can we give a person or an individual the blood groups. So we check the surface antigen on the RBC. Then we have antibody. Now if you try to recollect what I said about the antigen and antibody, I said that the body will not produce antibody for an antigen which is already present in the body. So if the body has antibody, let us say anti-A. If I say the body has an antibody of anti-A, it means this antibody can destroy any antigen which is named A. So this anti-A can go and destroy this antigen. And if an individual has a blood group A, it means he has antigen A. And if the antibody is against this antigen A, then the person will die due to clotting or due to any disorder. So the person having blood group A will have an antibody of anti-B. It means that if you transfuse a blood of any person with blood group B to a person in blood group A, then the blood will clot. Similarly, the person having group B will have anti-A, because it cannot have any antibody to the antigen which is already present inside this. Now, blood group AB has both A and B antigens. So, blood group AB does not have any antibody. And blood group O has no antigen, so it has antibody against both anti-B and anti-A. Now, why it has both? Because it does not have any antigen. So, if there is another person's blood coming inside, the body can respond to it as a foreign particle. So the body has the capacity to destroy the blood of any individual that is coming to the person. Now the question is, who can donate the blood to the individuals having blood group A? So we know that same blood group persons can give the blood to each other. So for example, if a person is of blood group A, so a person of blood group A can donate the blood to a person of A. Who else can donate? To check who can donate to whom, we have to check donors, antigen, and we have to check recipients, antibody. If both match, then no transfusion. This is the main concept that we use to determine if a person can donate blood to other person or not. So we have to check the donor's antigen and we have to check the recipient's antibody. Another concept to be understood is that if we take blood from an individual to give it to another person, we have to remove the antibodies. So as we have spoken, we usually inject the serum to the person, or we usually inject the blood which does not have the antibodies. So the antibody of the donor becomes insignificant. So we have to check the donor's antigen and recipient's antibody, so that if they both match, then we should not transfuse. Now what is this concept? Let us understand. Suppose a person having blood group B is giving the blood to a person having blood group A. The concept is B is giving to A. Now B has antigen of B from here, and B also has anti-A antibodies. Now individual who is B, he has antigen A, and the individual has antibody of anti-B. We saw that we have to check donor's antigen. This guy is donor, we have to check only antigen, then remove the antibodies. We have to check the recipient's antibody, then remove the antigen. If they both are matching, then we should not donate blood. Why? Because the person who is A group, he has abundance of anti-B, and if you inject any blood group which has B antigen, then this person's body can destroy this B's person's blood. So it may cause blood clotting. That is why a B individual cannot give the blood to an A individual, because if their both blood transfuses, then there can be a chance of clotting due to the antibodies present. So B cannot give blood to A. Can AB give blood to A? No. Why? Because if you see AB, it has antigen B and antigen A. Antigen A will do no harm, but antigen B is present in AB group. So if A recognizes antigen B, it will go and kill it. So there is no point of transfusion of blood. Now blood group O. Blood group O does not have any antigen. So it has no antigen. If there is no antigen in blood group O, B, the person who is individual A, he has only anti-B antibodies. And this blood group O does not have any antigen. So it can be easily transfused. So a person with individual who is having blood group O can receive blood from both A person and from O person. Similarly, if we take a person having blood group B, he can receive blood from B and O. A person having blood group AB, this is a very special case. Now, if there is a person who is receiving blood and who is AB, this guy will have no antibody. So whatever blood you give to it, whatever antigen the blood you give might have, this will not show any response. So AB can receive blood from every individual. It can receive blood from A, B, AB and O. What about individual O? If a person has blood group O, he has antibodies for anti-A as well as anti-B. So whatever type of blood group you insert, if you insert a blood group having A, this has antigen A. So this person can destroy this antigen A. If a person B is giving blood to O, he has anti-B. So this person can destroy this antigen B as well. If you give AB blood, AB has both antigens and this has both antibodies, so both can combine and destroy it. O has no antigen and there are only antibodies, so these antibodies don't have anything to attack onto. So a person O can only receive blood from O individual. So because of this table, we can say that the O person blood group, the person who has O group, he can give blood to all the individuals. So O is called as the universal donor. This is universal donor. And if we see this person who is AB, he can receive blood from all the 4 types of blood group. That is why AB is called as universal recipient. AB is universal recipient. So whenever there is a medical emergency and blood needs to be transfused to the person who is involved in accident, and if we do not have the time to identify his blood group, then what the doctors usually do is they just inject the blood group O. They inject the blood of a person who has blood group O, because O is a universal donor which does not have any antigen. So whatever the blood group of the person may be, it will not destroy or it will not collide with the person having blood group O. So this was the main concept of this was the main concept of ABO grouping. Let us sum it up a little bit and quickly. ABO grouping involves the grouping on the basis of the alphabetical characters, that is A, B, AB and O. This naming is based on the types of antigens which are present on the RBC surface or the surface antigen. If the surface has antigen A, then the blood group is called A. If it has B, then it is called B. If it has both the antigens A and B, then it is called as AB, and if there are no antigens, then it is called as group O. A person having antigen A or a blood group A will have antibodies against B antigen. A person having individual having blood group B will have antibodies against A antigen. If someone has AB group, they will have no antibodies, and if a person has O, they will have both the antibodies against both of the antigens. Now, A can receive blood from both A and O, B can receive blood from both B and O, AB can receive blood from A, B, AB and O. That is why AB is called as universal recipient, and O can receive blood only from O, but O can donate blood to everyone. That is why O is called as universal donor. How can we check if a person can donate blood to someone or not? We have to check the donor's antigen and we have to check the recipient's antibody. If they both match, then there is no transfusion. Then you should not transfuse the blood. If you transfuse the blood will clot and the person will die. So this was the first grouping. The second one is the RH grouping, that will describe or determine if a person will have positive or negative. So let us see. So now let us move on to the second type of grouping that is present in humans and other species. We will discuss that. That is called as RH grouping. Now RH blood grouping will describe if a person will have a positive blood group or a negative blood group, because we usually see that people have A positive or B positive. Some people have A negative, B negative. So we have understood the concept of AB, AB and O, that how do we determine if a person has A, B, AB or O. Now we have to understand how can we determine if a person has positive blood group or a person has a negative blood group. For that we will be learning about RH grouping. Now first we need to understand what is RH, and for that there is a very interesting story behind. First, before moving on to the name, there is one thing which we need to know is that RH is nothing but another antigen. So what is an antigen? Antigen is a substance, or antigen is a point on the cell which can determine what kind of cell is this, or antigen becomes the identity of the cell, or identity of wherever it is present. So RH is an antigen. If this is our RBC, so RH can be an antigen which is present on the RBC. Now let us move on and let us understand what RH is. RH is derived from rhesus monkey. So the individuals who discovered the ABO grouping, afterwards they tried to shift their study on different species to identify if they also have different types of blood. And there is a species, or there is a type of monkey which is found on the streets of some other countries, and that kind of monkey was named as rhesus monkey. This was a species of monkey. Now this rhesus monkey has something unique, as a new kind of antigen which was present on the RBC of this monkey, and this antigen was named as RH antigen. After studying ABO grouping, the scientists went on and researched on other animals like the rhesus monkey. They found this RH antigen and they were more curious and they were more interesting, and they wanted to identify, do humans also have this RH antigen. Then from humans they went to animals, and from animals they came back to human to check if human also has this RH antigen, and it was found that 80% of human have this antigen. So 80% of the human population has this antigen. So the people having the RH antigen are known as positive people. I mean, not positive people, they have the plus sign in their blood group. And the remaining people who do not have this antigen are called as negative sign people. So if there is a person who is A positive, it means on his RBC there is an antigen A and there is also antigen RH. Now this individual has a positive blood group. But if I talk about A negative, then the RBC of this person will have A antigen but there is no RH antigen. So this is A negative and this is A positive. Now we understood how can we determine plus or minus sign to a blood group. Now there is one interesting concept about RH. We have learned in ABO grouping that if I am of blood group B, then I have antibodies against A. We have anti-A. So the question comes, the person who do not have RH antigens, do they have antibodies for RH antigen? Now this is a very interesting case because they do not have. The RH negative people do not have anti-RH antibodies naturally. So naturally, if you check the plasma of any RH negative individual, you will not find the anti-RH antibodies. And this is a very important statement because this statement will be used to study a medical case in the upcoming videos, in the next video. So RH negative people do not have anti-RH antibodies naturally. If we give RH positive blood to an individual who is RH negative, if we donate the blood or if we transfuse the blood of RH positive individual to RH negative individual, then this RH negative guy will produce anti-RH. So only after the transfusion, if by any chance if the RH negative individual gets the blood of RH positive, then the RH negative individual will start preparing antibodies for the RH antibody. So this is the main base of what is RH grouping. So from this we can understand that a person who is having antigen B on a surface and who has RH antigen on the surface, then this person can be called as B positive. And if there is a person who has B on the surface but no RH, then that person can be called as B negative. So this was the main idea about the blood grouping. Now if there are many questions that which blood group has the maximum amount of antigens. So maximum amount of antigen is present in AB blood group. AB blood group will have antigen A, antigen B and also RH, not AB positive. It will have antigen A, antigen B and antigen A, antigen B and RH antigen on them. This is the maximum number of antigen that can be present in a individual with AB positive. Now, if you check the blood group of O negative individual, if I talk about O negative, so and also O positive. A person who is O positive, we have seen that this person will not have any antigen. But if a person is O positive, then this guy will have RH antigen. An O negative person does not have any antigen. So since there is no antigen, if we donate the blood of O minus to a person who is RH positive as well, so RH positive, the person who is RH positive, he will automatically not have any anti-RH antibodies. So O minus is actually called as the universal donor. So we know that O group is the universal donor, in that O negative is the universal donor. And AB positive is the universal recipient. So this was the main concept. Now we will try to understand about a medical condition.